Hello everyone and welcome back for more Intro to Biotechnology. Last time we got a head start on figuring out exactly what biotechnology is, so in this video we are moving on to the next section. We're going to start looking at the variety of different biotechnology products, and we're going to get a glimpse into some of the techniques for producing said products that we will be getting into much later in the semester. So, the first thing we want to do is differentiate between what are called natural products and then what are called bioengineered products. So natural products are molecules and substances that are produced, as the name indicates, naturally by organisms, and we can identify through research natural products that have a potential benefit to us as people, right? So a very good example of this historically is antibiotics, so penicillin being the best example of this. So penicillin is a compound that is produced by a specific subclass of mold-forming fungi called penicillium. It was isolated and identified way back in 1928 by a Nobel Prize winner by the name of Sir Alexander Fleming. And what he noticed is that any bacteria that happened to be in the vicinity of these mold-forming fungi were basically killed off, whereas bacteria that were much further away from the fungi actually managed to survive. And based on that observation, he hypothesized that the fungi are producing some kind of substance that kills off the bacteria, and of course, as the story goes, that substance ended up being penicillin. So antibiotics like penicillin, obviously very highly effective at killing and stopping the growth of several different types of harmful bacteria. And the idea here is that once we identified this, kind of the idea of what biotechnology was trying to do is twofold. Number one, we try to figure out a way to get these fungi to make a lot of these antibiotics, so not just the quantity that they make naturally, but we want to, again, manipulate and trick them into making lots of these antibiotics. And then the next part kind of gets into the bioengineering thing that we will talk about on the next slide. Another thing that we try to do is we try to take what we know about existing antibiotics and make modifications to the chemical structures of those antibiotics to make new antibiotics that maybe get around the troublesome ability that bacteria have of developing resistances to certain antibiotics. So, very good example of a bacterial infection that is all about antibiotic resistant is MRSA, methicillin-resistant Staphylococcus aureus. So resistant to most types of antibiotics, so it really takes a very specific type of antibiotic to get around that resistance. And as we are going to see here on this slide, so you'll want to, when you get your hands on these slides, you'll want to click on that YouTube video and actually watch the video that accompanies this. But basically, this is a video that links to the results of the Kirshany lab from the University of Harvard Medical School. Basically, what they're demonstrating here is just how rapidly bacteria develop antibiotic resistance over time. So watch the video, but I can go ahead and kind of describe what's happening here. So we have this kind of rectangular petri dish that bacteria can grow on. And when bacteria grow, they tend to kind of spread out from an epicenter from where you kind of plate them. So what we have here on the far left and the far right side are kind of the medium that the bacteria grow on with no antibiotic present. So that's what the 0 indicates. So initially the bacteria are plated on the far left and on the far right, and then as they grow and divide and spread out, they will spread from the left to the right and from the right to the left. Well, at this kind of interface right here, you can kind of see this little line right here. This segment of the plate and this segment of the plate that says 1X, that medium is impregnated with exactly enough antibiotic that should be sufficient to kill off these E. coli bacteria. So as soon as the bacteria kind of hit that interface between the 0 and the 1, they should stop growing because the antibiotic is effective on those bacteria and prevents them from growing. Well, as you watch the video and you see the time lapse occurring, you're going to notice momentarily those bacteria stop spreading, but eventually small little colonies here and there along the vertical axis between the 0 and the 1 will overcome the antibiotic and start spreading out on their own. So you can see that over the complete time lapse, as we go from no antibiotic to 1 times effective antibiotic, and then 10 times that concentration, then 100 times the 1x concentration, then finally 1,000 times concentration. The 1,000 times meaning this is 1,000 times the concentration of antibiotic that should be sufficient to kill off these bacteria. If you give the bacteria enough time, they spontaneously come up with mutations to their DNA. They basically figure out ways to survive in spite of the conditions becoming harsher and harsher and harsher over time with respect to the antibiotic concentration. So the whole point of a demonstration like this is to convince you that whatever our current cache of antibiotics we have currently available to us right now, it's not going to be enough. Bacteria are capable of developing resistances to antibiotics so rapidly that in biotechnology, our development of new antibiotics needs to at least happen as quickly as these new resistances happen. So very important application for what we're going to talk about here, bioengineered products. So bioengineered products really refer to products that do not necessarily occur naturally. So the products may be based on something that did occur naturally, but when we're talking about bioengineering, our ability to make a lot of a certain product or a modified version of something that occurs naturally is all going to be the result of biotechnology innovation. So like I was indicating, many of our antibiotics and other types of medicines are not really things that actually occur naturally. They're actually based on kind of ingenuity and bioengineered based on our need for these products. So we need lots and lots and lots of different antibiotics that are effective against lots and lots of different types of bacteria. There may not be enough naturally occurring antibiotics to satisfy that demand. So that's where bioengineering comes in. So another way of doing this, of producing bioengineered products, is by creating what are called genetically modified organisms, or GMOs. So just naturally, you probably hear this term most frequently when you're talking about agricultural stuff. So hybrid grown tomatoes and things like that. You hear GMO this, GMO that, and GMO is kind of a term that gets a negative connotation in the media, and really for no good reason at all. I think a lot of people, maybe the uninitiated, are just maybe a little bit afraid of what they don't understand. There really is no need to fear genetically modified organisms as long as the biotechnology is sound in what we're trying to do. So a GMO is an organism that has had its genetic code manipulated in such a way that this organism now makes a new type of protein that they did not make previously. They're able to make this new protein because they have acquired a new gene or a modified version of a gene that they already had. So let's look at an example of that. So fireflies, hopefully you've been able to see fireflies before. They're really nice to look at on kind of a hot summer night. So you see fireflies kind of buzzing about in the dark, and every now and then they will glow. Well, fireflies, their ability to glow is based upon an enzyme, a protein that they make called luciferase. So luciferase has a substrate that it works on called luciferin, and when luciferase oxidizes this luciferin, a photon of light is emitted. So this is, every time this reaction happens, the firefly is going to glow. So the idea here is that since luciferase is a protein, and since there is a gene, a DNA sequence that provides the instructions on how to make luciferase, well, theoretically, we can isolate this gene and transfer it to a different organism and ipso facto give them the ability to glow. So in this diagram that you're looking at here, If we can isolate and take the luciferase gene and transfer it to something like a houseplant, you can theoretically get that house plant to grow, or to glow, excuse me. So once you've done that, the plant is now what you would call a genetically modified organism. The natural or wild type version of that plant should not have the ability to glow, but because of our manipulation of genes, we can actually get it to do so. So let's look at another example. So this example is not only going to kind of back up what we were just talking about with bioengineering, but it will give us a little bit of a better idea for what the step-by-step methodology is here. So this time our gene of interest is what is called tissue plasminogen activator. It is an enzyme that is very effective at dissolving blood clots. So in a medical setting, you can definitely imagine a lot of utility for something like that. So tissue plasminogen activator, just on its own, is a natural product. So cells in your body do make this protein, this enzyme, but they don't make very much of it. So it's kind of the same issue that we ran into in the last video with insulin. A normal person should make insulin, but they don't make very much of it because insulin is already very effective at low concentration. So how exactly can you get a person to make enough insulin to warrant taking it and harvesting it? So we talked about the ethical concerns and why that's just not a very efficient way of going about things. So we're dealing with the same sort of issue here. This time, instead of insulin, we want this tissue plasminogen activator. So this is the basic methodology of how we can create a genetically modified organism that should produce a lot of this protein for us. So since the instructions for making this protein are in the DNA, we have to start with the DNA. So the first thing we have to do is identify on which human chromosome is this gene located. Okay, easy enough. Well, the next thing we need to do is to isolate this gene and to make many, many copies of it. So we're not actually going to cut this gene out of human chromosomes. What we're going to do is we are going to utilize a methodology called polymerase chain reaction, or PCR, to make many, many, many copies just of this one gene. We're not making a whole bunch of copies of the whole chromosome. We're making just a bunch of copies of this one particular gene based on having that gene already sequenced prior. So all you need is certain materials that we will talk about later on when we actually talk about polymerase chain reaction in detail, but you need a source of DNA. So you could say get it from someone's cheek cells. You could just swab the inside of their cheek and get some DNA that way and use that as a template for making lots and lots and lots of copies of this gene. Okay, so next we are going to take that PCR product, take the amplified tissue plasminogen activator gene, and we are going to insert it and ligate it into a vector. So a vector is usually just a circular piece of DNA that has a little empty spot that is meant to accommodate the gene you're trying to insert it. So the circular piece of DNA that contains the TPA gene in this case is called a plasmid. So the plasmid is then going to be what is introduced to what we are going to then call our genetically modified organism. So this plasmid vector is either transformed if you're talking about introducing it to a bacterium, or transfected if you're talking about introducing it to a eukaryotic organism. In this particular example, we are using Chinese hamster ovary cells, or CHO cells. So those are eukaryotic cells. They're much, much, much bigger and more complex than bacteria. So in this case, we would call this a transfection, basically tricking these cells to take up this plasmid that contains the TPA gene. And as we mentioned in the last video, DNA is universal to all organisms, so that DNA will be readily accepted, and we can get these CHO cells to start manufacturing our TPA protein based on the genetic information that we introduced. We give these cells the blueprints, they read the blueprints, and they make our protein for us. Again, we are manipulating them to do what we want. So this is our new genetically modified organism. These CHO cells are now what we call recombinant, meaning that they make a particular protein. They have recombinant DNA. They make a particular protein that they would not ordinarily. So we can basically maintain and propagate this line of recombinant CHO cells for a very long time, and all the while, as long as we're doing what we're supposed to, we can harvest and purify this TPA for medical use. It's really quite slick. So something that we should not overlook that makes something like that possible is that we have to know the sequence of a gene before we can really start messing with it too much. So in the previous example, and again with the insulin example that we talked about in the last video, the only way we're going to be able to manufacture large quantities of TPA or of insulin is if we know the DNA sequence of those genes to begin with. If we don't know the sequences, we can't possibly use PCR to amplify it. We can't possibly know how we're going to insert it into a vector. So we've got to know the sequence of these genes. So human chromosomes, which are basically the cookbooks that contain all the recipes, right? They are the compendiums of all of the genes that we have in our bodies, roughly somewhere between 20 and 30,000 different genes in the human genome. So if you kind of think about this for a bit, theoretically, we should be limited to manipulating through biotechnology only the genes that we have fully sequenced. And with 20 to 30,000 genes, kind of hard to imagine that we've got all the information on all of those, right? Well, think again, because one of the most important breakthroughs of the century, really, was the completion of the Human Genome Project back in 2003. So we have the entire sequence of the human genome, and that includes all of the genes. Now, to clarify, we don't exactly know what every single gene does exactly. We don't know what all of them do, but we at least have the sequence for them. It's like having a cookbook full of recipes that you've never tried before. You don't know what's going to happen when you actually make the recipe, but you've got the information if you want to try it, right? So because of this, we now have the ability to make all kinds of different recombinant organisms based on the need or desire we have for any of these 30,000 roughly human genes. So if 10 years down the line, we identify some gene that we need to figure out a way to make a lot of this particular protein product of this gene, well, we've had the information on how to make that and how to work with it ever since the completion of the Human Genome Project. So if you kind of put all this information together in kind of a biotechnology timeline, really because hopefully you've noticed how important DNA is to everything, really none of this could have possibly gotten started until we learned as much as we possibly could about DNA and particularly human DNA. So if you date back to the 50s and 60s with Watson Crick and Rosalind Franklin's breakthrough in discovering the structure of DNA, we kind of start to figure out how to work with recombinant DNA in the 70s, making genetically modified organisms in the 80s and 90s, so you can kind of see how some of the earliest advances we made in biotechnology really was all just about our ability to learn more about what DNA is, what it's made of, what it does, and what it's capable of, right? And then some of these later advances, such as coming up with an enzyme that will actually give genes that stonewash look, making insulin, making all kinds of antibiotics and medicines, really those are just kind of natural advances that are based on discoveries that we made many, many, many years ago. So kind of the thing that the book puts forth is that the biotechnology field really snowballs. So the more and more we discover, the more and more that we are capable of doing in getting organisms and cells to be manipulated and to do the sorts of things that we want it to do in order to make our lives better. All right, so that's going to do it for this particular video. So join us next time and we will continue to look into the biotechnology field a little bit further and we'll learn much more about it. So thank you for your attention and I will see you next time. Bye-bye.